And then the other thing is I'm going to do my best not to showcase my lack of knowledge when it comes to orchids. I appreciate all of your input and don't have a problem with stopping me and telling me the what for. All right. So just a little bit about me. I grew up in Phoenix, Arizona, born and raised. I went to ASU, where I got an undergraduate degree in zoology. While at ASU, I started doing some research with a gentleman by the name of Stan Faith, and he was interested in grasses and fungi that actually grew in the seeds of the grasses. And so a mature grass, an infected grass, would give rise to infected seedlings. And so this fungus was passed through the seeds through the years. And it turns out that this fungus had a very important role to play. And I'll show you that in just a second. So before I go here, has anybody not heard the term microbiome? Okay. A microbiome is a collection of microbes that inhabit some particular niche. So what I'm going to show you is that you have a microbiome, you all have a microbiome, and there are distinctions between all of your microbiomes. Okay, so I'm going to show you that. But for those of you who are a little queasy around microbes or not so interested in learning about them, you might want to consider this. Oh, so let me finish. I got my master's and PhD in plant pathology at the University of Kentucky. And this is when I started working with these fungi that grow inside of grasses. These are very, very important fungi because they produce defensive chemicals. Okay, so they grow in the above ground tissues and they produce what are called alkaloids. And these alkaloids are strong enough to kill grazing vertebrates or insects. So they're very defensive to their host. And this is very likely reinforced by this seed transmission, right? Generation after generation, this fungus is associated with this host. And so it's come to defend its host. It can protect it against insects. It can protect it against drought. And so I left with a PhD and I went to do some work with a true pathogen. This is called Alternaria brassicicla. It's a pathogen of crucifers. So things like broccoli, right? Everything that's associated with the broccoli plant and the model plant, Arabidopsis. So that was just a project where we were trying to identify genes in the fungus of this pathogen, this disease-causing microbe. Could we knock out a gene and stop it from causing disease? That was the role of that project. The truth is, not until my last month there did I finally knock out a gene, meaning I disrupted it in the fungus, and it could no longer cause disease. Okay, so part of my work there was it was oriented towards maybe GMO sorts of property where you could design genes to go into the host that could help it in that regard. For the last 15 years, until 2021, I was a principal investigator down in Ardmore at the Noble Research Institute. It's a plant science research institute. It's independently funded, wonderful place to do science. And so when I was there, I started working on root-associated microbes and whole microbiomes for plant improvement and soil regeneration. So that's kind of what I'm going to tell you about. And now, for those of you who might be microbial squeamish, you should know that there are at least as many microbial cells that make up your body as there are human cells. Okay? You're just as much microbial. Yes, sir.
On the skin as well.
Both.
Okay. So
the large, well, and I'll get to this. In terms of genes, the microbes give you 99% of your genes. Only 1% are considered human genes. So what that means is all of us are very, very impacted by our microbiomes. So if we look at how many microbes you have on and in your body, it's roughly 6 pounds of microbes. The large majority of these are in your gastrointestinal tract. These are the ones that are necessary to digest your food and assimilate the nutrients that are inside. But that's not all there is. There are actually microbes on and in most tissues of your body. even in your brain. Supposedly the blood-brain barrier was this great sort of thing to keep everything out. It's not sterile. There are bacteria in your brain, and they're not doing anything bad, and they may be doing something good. And I'll show you why we can say that. It's so important then that you have something called an enteric nervous system or a gut-brain axis that allows your stomach and your brain to directly interact and signal with one another. So much so that the brain sends down 500 million neurons to surround your stomach, your digestive, your small and large intestine. It's constantly monitoring what's going on in your stomach. Dysbiosis, meaning something that's not healthy, causes problems in your gut. And now we're learning that those gut problems can be linked to mental states. So my point being here is that they have different roles. They train your immune system in utero. So if a baby is born vaginally, it has one type of microbiome. If it's born by a C-section, it has a completely different microbiome. Some of those microbes it's missing are to train the immune system against things like asthma, right? These are some of the sorts of issues, bronchial issues, other sorts of things, even serious mental disorders, such as bipolar disorder and just a plethora of neurological defects. So we have the seed transmission of a vaginal versus a C-section. And if you've ever noticed, a lot of doctors giving a C-section will give a vaginal swab on the baby's head. Oh. What's that? Well, that's true. The dads who are in there with the mothers don't want to see anything like that. It very much is. The fecal microbiome is one that we've made the most progress with. And that's because of these gastrointestinal irritable bowel syndrome. There's now microbiome treatments for irritable bowel syndrome, right? So we're moving beyond this correlation stage to causation. That's what we need. Are the microbes driving the disease, or is the disease driving the microbes? Okay. All right. So what I'm talking about is a paradigm shift. Okay. You cannot separate the macroorganism from its microbiome. I would go so far as to say the future of medicine will be twofold: your genome, personalized medicine, and your microbiome. Those two will go hand in hand. Okay. Your genome is the microbiome plus your human part. Okay. All right. So my whole program is based on the premise that similar things occur in plants. Okay. The reason I say that is because there are microbes that exist on and inside plant tissues. The inside ones are called endophytes, which literally means inside plant. They get recruited from the air and the surrounding soil. Soil is the most biologically complex substrate known to man. In one handful of soil, there are literally billions of bacteria, millions of fungi, and hundreds of thousands of other sorts of things like amoeba, like protists, and different things like that. So very, very complex. But again, these provide essential functions, at least the ones that are endophytes, and I'll show you why. They provide functions for the host in exchange for carbon that the plant makes from photosynthesis. Okay, so the plant's fixing the carbohydrates, right? Turning light energy into chemical energy, and it's exuding it from its roots to pay microbes to find nutrients for it. Okay, so that's the symbiosis. Carbon for nitrogen, carbon for phosphorus, carbon for potassium, for micro, macronutrients. Why? Because microbes are really good at getting them. Okay, so so we get transfer of nutrients from soil to plant roots for uptake. And we do have a few that are well known, I'll show you. But again, they alleviate plant stress, some do. Improve resilience, some do. Some prevent bad microbes from causing disease. Some do, just like us. And just like us, there's seed-transmitted microbiome. Okay, so again, this concept of microbiome is a holistic one. But despite the sort of impact that using microbes in agriculture could have, we have no idea what the large majority of these things do. There are a few that we do know. This is a bacteria that infects the roots of legumes. Okay, anybody know why people like to have legumes? Grow legumes? Guess where that nitrogen comes from? These bacteria are capable of pulling it out of the air and fixing it into a biological form. Absolutely. Absolutely. These are actually structures that the host built. Okay. And it turns out that these bacteria are in the soil. They're recruited to the root. The plant starts making these little structures. The bacteria get inside and subsequently lose the ability to go back into the soil. And they start fixing nitrogen for the plant. And the plant keeps feeding it. The reason these are pink is because the bacteria uses hemoglobin to tie up all the oxygen and make it anoxic. Oxygen kills the fixation of the nitrogen. So it's got to keep that oxygen level very, very low. Okay. This is a fungus. It's called arbuscular mycorrhizae. It's a type of mycorrhizae in general, means fungus root. So these are all associated with plant roots. This one is a very ancient fungus that actually, it turns out, likely facilitated the colonization of land by plants. Okay. Before any plants got on land, fungi were there. Bacteria were there. Why? Well, fungi can decompose rock. Fungi can grow in a variety of very harsh climates. So they go up, they start turning rock into soil. And so the very earliest algae that we think probably gave rise to land plants was associated with these. And if we look back at the oldest fossils that we can find, if you look in the roots, you can find these structures. Okay. Another one, this is ectomycorrhizae, another type of mycorrhizae that occurs on forest trees. Okay. So temperate and boreal forests, this thing grows around the roots, right? And forms a complete, what they call a sheath, meaning any nutrition that comes to that plant goes through that sheath. That's correct. That's correct. So we're getting to where I am interested. I talked about the grass endophytes that I worked with. This is one that if you look at the grass, you can't see. If you look in and you stain the grass tissue, right, the above ground tissue, see these little squiggly lines? That's the fungus. Can't see it. It's inside the plant. But grazing animals, like cows that are fed on exclusively on infected grasses, will start to they'll lose their ability to thermoregulate. They'll spend all of their time in the water. If it gets severe enough, they'll start experiencing gangrene. They'll lose hoofs. They'll lose tails. They'll have stillbirths. Another class of these compounds from a different one of these guys is predominantly in New Zealand, which have a big sheep industry. Sheep feeding on a different grass with one of these start to experience what's called ryegrass staggers. I saw this in person. Basically, sheep walk along and everything looks good. If you scare them in any way, they go into epileptic fits. And they get stuck. They get stuck in fences. They get stuck all over, and it's a major loss to the industry. Again, a plant microbe association. This is my group, Microbial Symbiology Group. This is the iteration that I had at the Noble Research Institute. And what we do, what I think we uniquely do, is we try to balance application and basic science. Okay? At the end of the day, if I can't use a microbe, I'm not interested in it. And I'll show you that. So our goals: culture, screen, and deploy. culture new microbes, new types of microbes. We focus primarily on fungi and bacteria from roots of field-grown crop, forage, and turf plants. Why? Because we're eventually going to screen these, and we're going to incorporate ones that we like into elite cultivars of these crop and forage plants. So the breeders are coming out with this. I'm going to enhance it with microbes. Okay, so these applied sorts of aspects include culturing. Yes.
So basically, we typically, from the layman, we consider fungi microbes in terms of either parasitic or symbiotic. Is there a third term, like just neutral, we don't know what it is, that can be used to explain this, because it's kind of vague sometimes.
Yeah. So what I can tell you is parasites and mutualists are both symbiotic. A parasite is symbiotic with a plant. That's how it causes disease. It either lands on a leaf and starts forming a lesion or whatever. That's considered symbiosis, living together.
Interesting.
Now, a mutualism
Okay.
is a relationship that's beneficial for both partners. A parasite is beneficial for one and detrimental to the other.
Okay.
Does that make sense?
Yes, that is very helpful. Yes.
Okay. All right. So we do a lot of things, and we're trying to get these things back into the field. So we use culture, we screen them for traits, which I'll show you. We test them for compatibility with our target plants. We do a lot of in vitro stuff. I'll show you delivery methods. How are we going to put this in the field if it works? How are we going to deliver this? How are we going to make it so that farmers or ranchers don't have to do anything extra, right? We want to keep it as close as we can to current agronomic practices, okay? In terms of basic stuff, again, this can actually all lead to intellectual property. Each of these microbes has that capacity for the university or for whoever. I'm the inventor on one of them. So you actually, I'll show you what that is as well. But then we use these other approaches. These are culture independent, and they're so-called omics-based approaches. So transcriptomics, what genes are being expressed, metagenomics, who's there? And we can even use it to say, what are they doing there? There's a lot of power in the technology now that drives DNA sequencing, RNA sequencing costs way down. So a machine that would have cost $10 million in 1990, when they started sequencing the human genome, I can now do on my benchtop and have it done at the end of the day. It is an amazing jump in technology. And to be honest, that's what's driving the microbiome work. Because now we can ask not just what if one microbe's there, we can tell everybody who's there. Okay, so populations of microbes. And then, so we use all of this sort of stuff that's more basic. So what I'm going to tell you about today are the more applied ones. Two quick stories. One about how we get microbes, how we screen them, how we decide which one to put back in the plant, and then how do they look? Do they look like they're doing anything positive for the plant, say in the greenhouse? Okay. Now, I want to say this up front. The ultimate disconnect right now in using biologicals is from the greenhouse to the field. Okay. Greenhouse, simpler environment. No other fungi or microbes. Field, more natural, but more complex. So we need to move up and down this scale. We can even put them just by themselves. One microbe, one plant. And we can watch them with a microscope and see how they grow. And I'll show you something like that as well. So I'm going to tell you about that. And then I'm going to tell you about an interesting microbe that we came across. And it turns out it's very closely related to fungi in orchids. Very common. And so this has unique properties, and I want to tell you some about that. Okay, so when I first got to Noble Foundation, I was told that we were being involved in a big consortium who were put together to find crops that we could use for bioenergy, for biofuels. Okay, why? So this is a huge collection of academia, private industry, government affiliations, and we were all tasked starting in 2007, went through 2012. Then we got funding 2013 to 2020. So almost the whole time I was at Noble, we were funded by DOE, the Department of Energy, to find crops that we could use to make biofuels from. Now, why do you want to use anything besides corn? This is all that's used right now. Why? Because it's easy to digest. It's all starch. Starch, microbes, you throw in some yeast, it'll tear it up. But what about the complex carbon that's in the green stuff? That's also available to make biofuels out of it, but it's not as simple, right? It takes more energy input. And so we need to use the land that we can grow corn on to feed people, okay? That goes on higher quality land, but it's resource intensive. This sort of stuff, we find things with fast growth rates can be grown on marginal lands. They're not competing with food crops. They're there on marginal landscapes, and that's a good thing. And we can grow them without almost any inputs. Those are the characteristics that we need to make this affordable and competitive with gas. Okay? It's all about getting that fuel out, and it's more difficult with green material on plants. In our area, switchgrass was named as a primary candidate, again, by the Department of Energy. I don't know if anybody remembers it, but George Bush actually mentioned switchgrass and making fuel from it. Yeah, that's right. And a lot of things, to be honest. There's many feedstocks, as you can see. So we focused on switchgrass. Why switchgrass? Because it has an amazing root system. Okay. The shoot system is 6 to 8 feet tall after 3 years. The root system gets this deep at about 5 years. Okay, now why is that important? Well, number one, it's a native C4 perennial here on the plains. This is one of the big four: switchgrass, Indian grass, big and little blue stem. Those are the four. So it was chosen by the DOE. This thing ranges from Mexico to Canada, right? It's got very low nutrient and water requirements. You almost just, if you can get it out there, it'll just grow. It's like a weed, but this kind of a weed. Now, why is that specifically important? Well, it means that we can grow a lot of shoot biomass, right? This is 40 dry megagrams per hectare. The root biomass is 8 dry megagrams per hectare. And for me, this is one of the big things. When we started working with switchgrass, I agreed, okay, biofuels are important. We can do that. But can we use a plant that can actually stabilize the soil, that can help to regenerate the soil? Something like that could definitely help. Okay. Why? Because we have erosion. This one's just around the corner from my house. Tons. You know, we had the Dust Bowl, for God's sakes. All of the topsoil is gone, right? This is in 1937. This is in 2011. Okay. This is the water table, precipitation levels. We know if you live here, it bounces kind of all over the place. But in general, we're going to start seeing more drought. That's a given. That's going to happen. So we decided we're going to go out and we're going to try to find microbes that we can do to maximize the biomass of this plant and to limit the nutrients that it needed to grow. Where did we go to collect our plants? Our switchgrass plants. When we want to enhance switchgrass, we go out and we find switchgrass and we sample that to put in our cultivars. Okay. So my team for 3 consecutive years flew up into Ponca City. We split in half. Half went to the Tallgrass Prairie Preserve. The other half went across the transect, across northwestern Oklahoma, including restored CRP land, pasture, fallow farmland, degraded and extreme habitats, including the Great Salt Flat that's in the Panhandle. There were actually switchgrass plants growing right on the margins of this stuff. So very interesting for us. Again, we sampled 3 times a year for 2 years, about 450 plants, non-destructively, okay? We take a part of the plant, part of the root system, part of the shoot system, okay? Then what? Okay, so we go out, we collect all of these plants, we bring them back to our lab, okay? And we want to get the microbes. So how do we do that? Well, we start off and we split each switchgrass clone, right? Each rammet that we brought back, we split it. Half of it goes into a field so we can maintain the plant diversity and study that if we want to. And half of it went straight into the lab where we would split the tissue into roots and shoots. Now, even though that says shoots, we did very little of that on switchgrass because the stuff is like bamboo. You will just break scalpel after scalpel after scalpel. So we focused on roots, which is great because we're mostly interested in nutrient relationships. So we actually took all of that material, say the root material, split it in half, and half of it was plated for fungi, like these cultures. Okay, these are in media in petri dishes. That's just a small sample, and half bacteria. And this is plated on a bacteria-specific media. Okay, we ended up getting about 555 fungi and about 528 bacteria. I should say, because I forgot, all of these root tissues are surface sterilized very vigorously. I mean, bleach, straight up bleach for a minute, ethanol for another 3 minutes. The reason why is because there are microbes on the outside and on the inside of the root. The ones on the outside include all kinds of saprophytes, cosmopolitan fungi like penicillium, aspergillus, they're everywhere, okay? And we know that there is communication between the plant and the microbe before the microbe is given access into the plant root. So by burning everything off of the surface, focusing on what we can get from the surface sterilized tissues, we're enriching for endophytes. And those are the ones that the plant has said, come on in, which would imply maybe they have a function that the plant wants or needs. Okay, so we take each of these and we take some DNA from them, and we can tell you who they are by sequencing a bit of their DNA, okay? It's actually a ribosome subunit. So for fungi, you use this subunit. For bacteria, you use this subunit. And they're very, very useful for classifying bacteria and fungi. You can get a very good idea of what you have, okay? All right, so what do we have? Well, this is an example. These are the bacteria, endophytes, that we grew out of switchgrass, okay? And we know, so we have Varioborvax, Klebsiella, Bacillus, Rhizobium, interesting, Burkholderia, Enterobacter. These genera, these genus, right? They are all known to have beneficial microbes in them, okay? That's a good sign. Of course, the big pie charter was Pseudomonas. That's just simply because Pseudomonas, again, is a cosmopolitan bacteria that's everywhere, okay? All right, so what now? We've got the taxonomic configuration of all of our endophytic bacteria from native switchgrass. Now, how do we choose from all? I can't take 600 bacteria and 600 fungi to the field. It's way too complex. So how do we choose is we screen them using high throughput assays that we've developed in our lab. And what we can do with these assays is start to assign traits to a given microbe. So with these assays, I can say, well, I'll show you, as a matter of fact. This is one assay. This is in a petri dish, okay? And this particular one uses an agar, a media that's called PVK media. What that means is that it has phosphorus, a type of phosphorus that makes the media cloudy, okay? Now, if any of that phosphorus in the media is solubilized, right, made available, there's a halo that grows around the microbe where it's solubilized all the phosphorus right around its colony, like this. See that zone of clearing? Zone of clearing, zone of clearing. All of these, and that's not the whole collection, it's just some of them, actually have the ability in a high throughput assay in our lab to get phosphorus solubilized. That's what they do in the soil, right? In the soil, phosphorus is bound to various things, and it's got to be solubilized to be given to the plant, okay? So that's one. This is another one. This is called a high throughput ACC deaminase active assay. Don't let that scare you. I'm going to show you what it is. It's very simple. When a plant experiences stress, whether it's heat or light or lack of water, it starts to respond by producing ethylene. Ethylene is a plant stress hormone, okay? So in general response to stress, it starts pumping out ethylene. Now, to do that, it's got to use an enzyme that converts this compound, ACC, to ethylene. So what we, as a response, the plant then senesces, it dies, other sorts of things when this plant stress hormone is there. So this enzyme we can block. So we can block ethylene production, okay? So we no longer get a dead or dying plant, and the plant behaves like it's not stressed, okay? Maybe not always a good thing, but the good thing about it is it's responsive to various types of stresses. It's kind of a general stress response because ethylene's a general plant hormone. Here's another one, auxin. This is a growth hormone that plants produce. Well, it turns out some microbes produce it as well. There's a disease called foolish seedling disease. This is a fusarium fungus that prematurely causes the rice to be produced. They overuse their growth reserves and die, okay? So what we have here then, this auxin is found in root and shoot tips, promotes cell division, stem and root growth. Again, here's no auxin. This is a defined concentration of auxin, and this one, a bacteria, is somewhere in the middle, okay? Auxin producers. We can test to see if they have the genes they need to take that nitrogen and put it in the ground, fix it from the air. And we can actually just use a very simple PCR to see if the bacteria have the genes they would need to fix that nitrogen. So these are strains that actually show that band. They have that gene. This is a positive control. This is a negative control. So we know that the size we have is very useful. We can take those ones that have the genes, right? We don't know if they're expressing them. We don't know if they actually have a function. And we actually use an assay where we can draw the headspace out of a plant growing in a tube, and we can detect whether or not it has the enzyme that can cleave a dinitrogen bond, which is what it would need to fix nitrogen. It's got to break that triple bond. And so that tells us then, not only does it have the genes, it's able to use it in a way that we want it to use it, okay? This is one for biofilms. Biofilms help bacteria stick to the outside of roots. They're kind of mucilage, sticky stuff. And in this assay, they turn purple if the bacteria produces them, okay? So again, each of these wells, we call them little wells, they're just little things. We fill them with the bacteria and we put in the substrate, whatever it is. And if they're able to cause this biofilm, so you can see the green here is all bacteria that are sticking to the outside of roots, okay? Same here, you can see them all over. This is an electron micrograph. Very easy to screen, high throughput. Here's another one for iron chelation. We don't have to go into it. This was a gentleman, he joined my program for the summer, and he actually helped develop these assays. The last one is a biocontrol of plant disease. What we do is we take our bacteria and we put it 3 places on the plate, and then we grow a pathogen, a fungus that causes disease, in the middle, okay? So this is our positive control. So you can see here, the fungus will grow all over the plate. You see here, bacteria have no impact. Bacteria have no impact. But can you see the reduced stuff here and none here? These bacteria and these bacteria, then we could try to use to suppress the growth, in this case, of cotton root rot. This pathogen is one of the most damaging to Oklahoma agriculture. Just because it says cotton root rot, it infects a lot of things and it kills them and it builds up in the soil, okay? All right, so at the end of the day, we have all these traits and we've given them to different bacteria, and we see if we can find ones that have more than one useful trait, okay? So in this case, if we look at this being whether they have ACC deaminase, this being whether they can solubilize phosphorus, this being whether they form a biofilm, and this one, whether they have the nitrogen genes, okay? So the ones that are in the middle of those have multiple traits. For example, this is 2 bacteria strains that have biofilm formation, phosphorus solubilization, and this one, ACC deaminase production, okay? We have more that have different groups. And here we have one that's got 4 different traits. That's a target. That guy's a target. If it does one of those functions, we're in good shape. All right. And then we have 45 isolates which didn't have any of these traits. Okay. So we don't get positives all the time. Then we take those ones that have the traits and we say, we're going to put them back in seedlings of our target host and see if they do anything. Okay. Again, high throughput. I can't wait for mature switchgrass plants. Okay. I got to do it as a seedling. So here what you see is all of the different ones that can solubilize phosphorus. Now, if we inoculate seedlings, we start off with 9. Then if we see a greater growth than the water control, like these, then we know it's got that trait and it improves growth. Okay? Same with ACC deaminase. Look at all these ones that improve growth over the water controls. Okay? So we created a simple community, a microbiome, and we included 3 of the phosphorus solubilizers that each promoted growth. We included 3 of the ACC deaminase, again, tremendous growth production. I should say the ones that look good with the 9 seedlings, we then test on 90 to make sure we get that phenotype, okay? These last 2 are the nitrogen fixers. Both had the genes. One of them was able to promote growth. This one actually passed that last assay where it actually had the enzyme to break the bond. This one had. So this was our simple group, okay? Now, I'm going to talk about this guy. How are we doing on time? All right. How many? 20. Okay, great. Okay, so this is my favorite microbe. I'm going to tell you why. So its name is Serendipita. Great name, you got to admit. Great name. And it's a fungus that grows in the soil, and it's related to the fungi we call wood rotters, okay? These are the brown rotters, the white rotters, and the soft rotters, and they're all what we call decomposers. Decomposers are responsible for breaking down the most complex carbon structures, cellulose, lignin, things that nothing else, no other microbes can mess with except for decomposers. So they actually lie at the top of the carbon cycle. And without their activity, carbon would become bound in unuseful forms, and we would get nowhere, right? So all the leaf litter would just keep building up. All the dead trees keep building up. Everything's going to build up because there's nothing to break it down. It's not like it breaks down by itself. It's decomposed, okay? Now, the fungus I just told you is part of this group, and it lies right here. This is a gene tree, and this tells you how these different groups are related. This is the one that I'm going to be talking about. It says here Sebaceanales. That is an old name. I imagine orchid enthusiasts know how often species names change. That's one. Now, I want to blow this up just a little bit, and I want to show you this group, one of the most fascinating things that I've seen. If we look at this group as a whole, each of these colors indicates what kind of mycorrhizae it can form. Okay, so this isolate is ectomycorrhizae from pine. Okay, ectomycorrhizae. Great. So what we got it from is what the genes tell us it is. Great. Okay, now, but what I want you to see is that within this relatively small group that came from that much bigger tree, we find isolates capable of forming every type of mycorrhizae except for the arbuscular type. the one I showed you in the beginning that forms the tree-like structure. The rest of them are formed by fungi like this. Now, why is that important? Because in each of these cases, the fungus actually is capable of decomposing organic matter that it finds in the soil, in addition to being able to infect a plant. Okay? In orchids, for example, and this is where I'm going to show the lack of my knowledge. Orchid mycorrhizae actually are required to get seed germination in the plant. Okay, I know there are new methods. I think we can get the seeds to imbibe some water and things, but in general, in nature, they require the fungus not only to grow onto its roots, but to give it carbon. Why? Because most seed, the fly seed, has no gymnosperm. It's got no seed reserves. It's got no starch. The only way it can grow is if something else gives it carbon. This is the guy that does it. And he does it by getting some of that organic matter that's in the soil, decomposing it, right? Soil particle is basically an aggregate of all nutrients that you would need, right? Macro and mineral nutrients, organic matter, all of that sort of stuff. So what we believe, and what it's been shown in orchids, is that the fungus actually starts off giving carbon to the host plant, right? Then if in this case, the orchid becomes chlorophyllc, can start fixing carbon, it will reverse the flow and the plant will start giving the fungus carbon. However, there are orchids that remain achlorophyllous their entire life, no chlorophyll, no green. Those types of orchids depend upon the fungus for their entire life cycle. They are called parasites. We call it mycoheterotrophy. They're literally feeding off the fungus. Okay? This is what one of these fungi look like. This is a petri dish. This is the fungus here. It grows in a circle. The oldest part is here. The newest part is at the edge. So it grows out like a circle, like a sending these filaments into all these regions. We can grow it in liquid culture. Great. This is what it looks like. So this is stained with a stain that's specific for fungi. Okay. And this may not mean a whole lot, but it's very beautiful to me, number one. And number two, it's got this very unique morphology that we don't understand. We don't know what it does. We call this a type of filament that grows bulbous for whatever reason, okay? And I'll show you why we think that might be important. The blue stain is on the nuclei. So every nucleus is stained blue. So you can see that one cell has several nuclei, and we can even catch them passing nuclei between the cells. All right, this is what it looks like. This is how infection proceeds. It starts off above the root cap into the region of rapid cell division. This is a root that's been cross-cut, and you can see these are the root cells. This is the very outer layer of root cells, and this is the fungus that, again, has been cut in half. So you see it like you're looking at it face on. It starts off in these outer parts of the root. As infection proceeds, the fungus starts moving interior. Okay? It never crosses the stele, right? The vascular bundle, but it starts packing into these cells like this. And those cells aren't dead. Those cells are perfectly alive. All of the green is the fungus again. The red stain is also nuclei, but most of it, the red, is the plant cells. So you can see it just literally packing in these living cells. No sign of disease, no sign of necrosis, no sign of any of that. So we decided, well, I want to try these things. Why? Because a gentleman back in 1986 decided that he was going to work with a fungus that he pulled out of the Tar Desert, and he found it had very unique properties, including an unlimited host range. He found that in most of those hosts, it would boost biomass and productivity. That got my interest. There were none other than that one strain, except for one orchid collection trip by James Workup, 1985 and 86 in Perth, Australia, outside of Perth. He had the only collection. There were 12 of these strains. of this type of fungus. That was it in the entire world. So I said, I want to get these strains and I want to test them. So we used switchgrass seedlings that we literally, this is very primitive, but at the time we would take this juice that's got the fungus in it and literally put it in these things and dip the roots in them. Okay. So just to put it on the roots, and then we put them in soil and we grow them in the greenhouse. So this is the control. We've separated the root system from the shoot system. Okay. This is the control. These are 6 of these orchid mycorrhizae. Root system, shoot system. Again, not an orchid. Switchgrass, not an orchid. Okay. All right. So there's our control. We started beefing up our ability to deliver this fungus, how we were going to improve on what we had done. We can't dip 10,000 wheat seedlings, right? Not going to work. So we started looking at different things. One of the good things was we could grow both the plant and the fungus on the same artificial medium. Okay, that is very useful for a lot of reasons. We also took sorghum grain and we sterilized it in an autoclave and we mixed in some of our liquid culture in there, shook it up and grew it for a couple of months. And you could actually see the fungus growing on throughout the sorghum. So that became our secondary type of inoculation. Now, there's a lot here. I don't want you to get too concerned about this. What I want you to see are, these are 4 graphs. This is, let's see, this is shoot length, shoot mass, root length, root mass, okay? Now, between the left and the right, the left side has normal water. The right side has restricted water, okay? Now, what I can show you here is the first, second, and third harvests at 2, 3.5, and 7 months. Okay, that's what we have here. So let's just say we're looking at this one. This is for root length, okay? No difference at any of these times. The root length was not enhanced. Let's see, but the root mass was crazy enhanced, right? So this is with the fungus on the left and without the fungus on the right. So first time frame, not much. Then the fungus starts making its plants outcompete. And again, this is in normal water. Look at the one at the 3rd time. It's absolutely massive. Now, if we look at the drought side, it's even more significant. Okay. So now we're looking at water restriction. Again, the same things, shoot length, shoot mass, root length, root mass. And again, without the fungus and with the fungus at the 3 times. All 3 times, very, very significant improvement in shoot length, in shoot biomass, and in root length, again, no big deal. Root biomass, very significant. The biggest thing I want you to take from this graph is if you look here, we have normal watering and the plants without the fungus, okay? Normal watering, plants without the fungus. If you look at this one, this is water restriction and with and without the fungus. So what you can say is that plants that have the fungus at all 3 time points and water restricted actually perform better than plants with normal watering without the fungus. Okay, so the presence of the fungus improves the plant even more when resources are restricted. All right. This is our latest delivery strategy. This involves bentonite clay. If anybody's ever tried to soak up oil on their driveway, this is the sort of stuff they use. Okay, it's a bunch of mineral oxides that have been fired to 10 million degrees, basically are just a substrate. You can't liberate any of these anymore. It's been fired. What we do is we take these things and we soak them with a broth that the fungus likes. Okay, and we let it take it all in. All right, and then we put some liquid culture in there, drink it all up, and we let it grow. Oh, now should be here. No, sorry. What it should show here is that so this was our kind of our gold standard. And so every few months, we would take some of these particles out and put them on a plate and see if the fungus would grow out. Okay, we got near 99% grow out at 5 years for a delivery particle that was just on the bench, not treated, nothing. Okay, that might be easier to get into an agricultural system, right? And they're very, very stable. Brilliant. Okay, so we decided what we're going to do is we're going to put these guys with our fungus. And at this point, we were done with the greenhouse and we were ready to go to the field. Okay, so this is how it worked. This is the first year results. Plant height, tiller number, tiller girth, and a measure of photosynthesis. Okay. Purple is fungus plus bacteria. Green, control, blue, just the fungus, and red, just the bacteria. So the combination does very well. Okay. Here, combination. Next, fungus. And then the bacteria is right with the control. Combination, just the fungus. Okay. The rest were just the control. This one, the measure of photosynthesis doesn't appear to be any different. But this is in a complex field environment. Okay. I can't tell you how important this sort of data is. This is at a second site, a field site. Okay. Just a little bit of an impact from the fungus. Again, the fungus is here, no impact from the combo. Fungus is here and nothing here. All right. So unfortunately, we were in the first year of our field trials when the Noble Research Institute decided to shut down their research division. So all the scientists were lost. All of the international postdocs had to go home. And it was pretty rapid. So unfortunately, we had to abandon our field trials. With a field trial, you need at least 3 years of data to convince anybody of anything. Now, here's the most amazing thing. The fungus that came from switchgrass, right? I got one out of the roots of switchgrass, not an orchid. And so this one, I actually got to name, okay? I named it after the DOE Biocenter that gave me the money to do the search. They were called BESC. Now, this is the only strain from North America and the only strain that's not from orchids, okay? Took me 1.5 years to get it out of the roots of switchgrass. I knew it was there because I could PCR it. It was there, but it took a lot of effort to get it out. Now, we've taken that single strain and inoculated each of these hosts, these with the improved biomass percentages. This is a crucifer, a model plant, Arabidopsis. This one doesn't form relationships with the nitrogen-fixing bacteria or the arbuscular mycorrhizae. We get a stable infection. That means that this whole signaling system is different, unique. Alfalfa, 50%. Medicago, that's a relative of alfalfa. Switchgrass, 330%. That was in our drought study. Winter wheat, 30%. Tall fescue, 37%. We've also done rice, corn, Bermuda grass, cotton, pecan trees. All of these we can successfully colonize. We haven't done growth studies with all of them, but we have done them. Right, winter wheat, this is my target. This is the one that I have been brought on to improve with microbes. Okay, that's everything. This is that fungus on the root systems of winter wheat plants. Duster, this is a genotype that was developed at Noble. So what you see here then are either the control root systems, these are very small seedlings, or the ones with the fungus. And what we've done is grown them under full nutrition, under nitrogen limitation, or under phosphorus limitation, okay? And we counted the number of root tips, all these different tips. And the fungus we put right in the middle. Okay? Now what you can see is that the number of root tips in full NMP it's better, but it's not significant. But look what happens when we limit nitrogen. And look what happens when we limit phosphorus. You can see it. You can see that these have more root tips than these. Now, what if we look at the root hairs? Okay, so not only do plants have roots, each of those roots has little hairs that come off of them. This increases the surface area that they can absorb things through. Okay, this is our control, winter wheat plants. This is our one with our new fungus, the one from switchgrass. This is one of the ones from orchids. Okay, dramatic improvements here, dramatic improvements. This is what the fungus, remember those bulbous hyphae? If you look in these root hairs, that's what you see. You see they're fully colonized and perfectly healthy. Okay, this is plants. This is in the greenhouse. Okay, again, full nitrogen and phosphorus, nitrogen limited, P limited, with the fungus, without the fungus. Okay, so again, full nutrition, limit either nitrogen or phosphorus, and it goes through the roof. This is what we want for low input agriculture. Okay, exactly this kind of a thing. And in fact, we've been able to put winter wheat in the field and generated an 18% gain in wheat grain yield. 18%. I don't know a breeder who could give you that. Okay, now, interestingly, these are just the particles. Okay, this is particles with the fungus. Just the particles give you a bump of 9%. But if you have the particles inoculated with the fungus, you get a total another 9%. Okay, very, very reproducible. We've started taking new approaches to deliver our fungus. One is peat. Everybody know what peat is? It's organic matter. Okay, again, we think this fungus has some decomposer activity, right? So peat could be a good seed coat. Okay, so we had surface sterilized seeds, seeds coated with canola oil, and seeds coated with peat that have the fungus. How do we know they have the fungus? See all the strings, all the filaments? Totally colonizing this stuff. Okay, 2020, our wheat trial at one of the farms at Noble. Again, we had seeds that were coated with just peat or seeds with serendipita. These we tried to sow at an industrial scale. Okay, so they were sown by a spreader in a decent sized plot. At our first collection, we found significant amount of the fungus on top of the seed. And again, we had to abandon them. Okay, so now at OSU, I'm tasked with trying to help different Oklahoma crops with fungi like this serendipita. Okay, these are my targets. This is number one. Number two, you know, everybody likes their turf grass here. I don't, you know, I'm from Arizona where there's like a gazillion golf courses. But again, pecan, that's another one. Pecan saplings typically don't become productive until about 8 years after planting. One of the most significant impacts that we see across the different plants we try to inoculate with serendipita is increased early development. So if we could turn around and say to pecan growers, we can get you maturity at 5 years or 4 years, that would be a major, major thing in the pecan industry. Okay, that's the sort of stuff that I'm interested in. Now, we are starting with our serendipita strain, inoculating all of these already. We've gone out into the field and started collecting our turf grass microbes. We're going this fall, we will start our winter wheat field trials. Okay, we're going to start new, and we're going to look for new things. So again, you want to improve wheat, go look in wheat. You want to improve bird Bermuda grass, go outside. Why? Because a microbe that's growing inside a perfect habitat doesn't need microbes. It only needs them when it's stressful. Okay, and that's what you find outside. Okay, it's got to live through the temperature cycles. It's got to live through the water cycles. So again, that's where we look. We also believe in local solutions to local problems. So if I'm going to improve crops here, I'm going to collect microbes here. Why? They're likely adapted to the climate, and because they're local, they're not restricted. We can put them back into the field. We can test them because they came from the soil. Okay. Now, here we are, Orchid Mycorrhiza. I'm sure you guys know this. These are the sorts of fungi that actually colonize the roots of orchids. Now, instead of the arbuscule, the tree-looking thing, or the bulbous hyphae, in orchids, the fungus forms what's called pelotons. Okay? These are hyphal coils, and again, they serve to increase the surface area that the fungus can send nutrients into the plant, and the plant can send carbon into the fungus. It's all about surface area. Same with the arbuscule, same with the hyphal coils, same with the bulbous hyphae. It's all about getting as much surface area to interact with the host, okay? Now, this is what I was saying before. Dust seed requires the mycorrhizae to germinate in nature, okay? Achlorophyllus seedlings, they actually require mycorrhizal-derived carbon to survive. Some of them stay that way, parasitizing the plant, and in a process called mycoheterotrophy. Others will start to produce chlorophyll, and they'll start to feed the fungus back. Now, here's some questions that I'm going to be posing to you here in the last minutes. One is, you obviously are experts in growing orchids and making new varieties of orchids. I wonder if growing them like this removes their microbes. Why? Because they're not in stressful environments. They're not outside. They're not dust seed. You're making clones, right? Okay, that's kind of interesting. This is how it works in nature when the orchid's out here and it's just starting off and it doesn't really have the ability to produce enough carbon. Not only does the fungus provide it carbon, guess where it gets it from? Anything around it that's producing chlorophyll. It'll simultaneously attach its filaments to the orchid and the tree. And it'll pull carbon through this tree to feed it to the orchid. Isn't that cool? I was like, wow. Good question. That is a darn good question. Hopefully, this thing's going to start photosynthesizing and return the favor. If it doesn't, it's a parasite, right? The fungus can't help it. It's being parasitized. Okay. Could studying mycorrhizal partners improve orchid conservation efforts? This is the main question I have for you all. I don't know the answer to this. I don't know if anybody knows, but I think it's something that we should consider. If we really want to preserve the diversity, right, their ability to continue to grow in a troubling climate, it's going to get more erratic. Maybe we should start thinking about collecting these microbes from the orchids as we get them from outside. Now, does anybody in the room wow orchids from outside? Anybody know where they grow? The reason I say this is because I think I would love to start cataloging these microbes. What sort of orchid they come from? If we look in similar orchids, do we find similar microbes? Do we find microbes that tend to be associated with plants that are growing in more sun than they typically would, or growing without more water than they typically would? When I go out, say in a wheat field, if I see a plant that's growing this much taller than the rest, I'm sampling that plant because it's possible that it's picked up a microbe that can improve its growth, okay? So these aren't currently preserved, but they could be. We could determine their taxonomy, or anybody, whoever you would like to work with. We could determine a taxonomy, and then we could store them in a culture collection, a national culture collection, actually international, called the American Type Culture Collection. And they store cultures just for this reason.
Oh, hey, Kelly, this summer, I saw some orchids in Grand Teton National Park.
Oh, really?
Yeah, I saw 3 kinds. I can send you some pictures.
Yeah, I'd love to see them. I'd love to see them. Thanks, Yuho. Then I start to worry about this. This is what I discussed earlier by asexually cloning versus seed propagation. I don't know. I don't know. It's possible that some microbes go through the protoform. They actually are maintained. We could easily take a little bit of DNA from a little bit of a root, and I could tell you what's on it or in it. And so what I'm wondering is, could we use this to improve reintroduction efforts, improve establishment, overall health, stress resilience? I showed you all these sorts of screens we could do for these microbes and figure out what their potential is. And then, we could even do re-inoculation experiments to study the biology of the interaction, right? So that is what I have for you. This is my group at Noble. We were, again, the microbial symbiology group. Wonderful scientists, wonderful place to do science. Notice, this is my current group at OSU. Yes, they look like lab dresses. I'm sorry, we just pulled them out of the packages, and only Layla was smart enough to put her hands in to hold it down. But this is my group. This is our department head, Dr. John Gustafson. Again, we're the Department of Biochemistry and Molecular Biology. This is my new postdoctoral scholar, Walid Asghar, my new lab manager, Jacob Swenson, and Layla Ismail, my new grad student. So with that, I would like to thank you for your time and invite any questions.
So in particular, we're talking about a plant in a sterile environment, typically indoors or something. There's no microbes because it's not necessary for stress. So the question becomes, if you even were to want to include the whatever in any kind of indoor, how often would you have to resupply them? I mean, typically we're talking about probiotics that they
Yes.
recommend taking on a daily basis to
Yes.
maintain the digestive system. So how often would you have to reintroduce them into?
This is a great question. And what I would reply is, remember, it's got decomposer activity. Okay. So it can exist in the soils without the plant if there's organic matter there. Now let's think about an agricultural situation. And let's say you're growing wheat. Every year, you're inoculating your wheat with the fungus and you're putting it in the field. Okay. That's the only organic matter going into the field. So what you're doing, it's kind of an early bird gets the worm. You're putting it out there, hoping that it's maintained. And what we found is through a growing season of winter wheat, the fungus survives. So we know that it's still active at the end of the year. So what I'm wondering is if we keep this inoculation going, eventually we're going to have a field that is super productive, whether we inoculate it anymore or not. Maybe.
The thing is
Yes.
with,
Oh, I'm sorry.
go on.
I have two questions. So I grow all my orchids indoors.
Okay.
And so as opposed to a conservation viewpoint, so what if I started inoculating my orchids with this Serendipita beskyii? Would that help improve the growth of my orchids? That's the first question. And number two, so what happens if I overwater, over-fertilize? Is that going to make things way worse then?
You won't need to water or fertilize with the fungus. It's not going to take additional water or nutrient resources as long as it's got an organic matter-based substrate. The first part about growing orchids like are currently being propagated, yes, I think there would be an impact. I think it would be more dramatic than you would see outside because we could control the inoculation, right? We can grow it in however you all grow it. If you have a variety of substrates, something that maybe is nutrient poor, nutrient rich, any of that sort of stuff we can test. And we can test right off the bat.